Hi, this is Sumathi. Today we have to discuss about the topic cell and molecular biology. Second name is zoology students. RNA synthesis. RNA synthesis is a very interesting topic. Before we have to know about the what is DNA, all the cell organelles also. So this module we have discussed about RNA synthesis. Normally cell also, central part also have the nucleus have the DNA have the genetic material that is called chromosomes, and the chromosome have the all the hereditary material transferred from the one generation to next generation. RNA synthesis also occurs from the DNA. That is a very interesting topic. Normal general tacoma, we have to explain how the DNA code for protein. The protein also very important. Why? Because the body filters also protein. All the activities in our body controlled by protein. It have the RNA synthesis have three regions or three stages also. That is called replication, transcription, another one is a translation. Once the DNA is replicated to strand, the information is copied into the RNA by the process of transcription, translation and protein synthesis. We have to know about the process of transcription. It is creating RNA molecules from the DNA template. That's very important. It is one of the complex process and also involved many protein, and also it is essential of all the organism. It's part of all the organisms formation of RNA synthesis. The first step of transcription is the gene expression that involves the formation of RNA molecule from the DNA. That is one of the important tacoma. Why the RNA synthesis is important? It is occurs in the cytoplasm and also formation of many protein. All the enzymes, hormones, everything also participate to formation of new things. That is a very interesting topic. First, we have to know about what is mean by transcription. It is one of the first process of gene expression. The genetic information flows from the DNA to protein, and the flow of the information will take place sequential process of transcription and translation. What is meant by sequential process? It continually occurs in the one by one. That is called the sequential process. Only one strand of DNA is copied. Not two. Normally DNA is a double stranded, but only one strand of DNA is copied. The process of transcription. The template strand and RNA synthesis is called mRNA. The template strand means new template strand. mRNA means messenger RNA. The main motive of the transcription, RNA synthesis from the DNA sequences. It does not from the anything. The RNA is normally transcribes and carries the information using to encoded of the protein. Why the encoded of the protein also important? All the parts. You know very well about all the parts in our body. For example, eye also have the different proteins coded, because eye muscles also very sensitive, and we have to know about the retina and everything, rod, everything also some visions. At the same time, our heart muscles are totally different from that eye muscles, because they do this different function. So the heart muscles are developing from that once it is coded of the different protein, eye muscles coded of the different protein. So the RNA is coded for formation of new cells, new protein in our body. RNA polymerase, it is one of the important enzyme involved in the transcription. It is uses of single strand DNA to synthesize a complementary RNA strand. Normally, the DNA dependent RNA polymerase binds to the promoter and catalyzes a function that is called polymerization. You know, the formation of any one of the template strand, it have the two N. One is a 5'N, another one is a 3'N. Once it reaches the terminator sequences, the process to terminate and newly synthesize the RNA strand, and also it is released in the cytoplasm. This is a very nice diagram to explaining the RNA synthesis. First one is a 3'N, another one is a 5'N. Normal first the pink color also creating the promoter. Another one is a green color also indicating the terminator gene. That is a template strand, means only one strand is called template strand. Another one of the 5' and 3', another complementary also sequences also formation. This is a strand also formation of RNA synthesis. The stages of transcriptions occurs in the initiation, elongation, another one is a termination. Everything also processed by enzymatically catalyzed the steps. First one is a initiation. The initiation functions next. RNA polymerase attaches the DNA molecule and move along the DNA strand until it recognizes the promoter sequence. Why? That is, a promoter sequence also initiates the next elongation function. So these are known as transcription starting site. After that, the DNA double helix and unwind. All the bases of each of the DNA strand, it is exposed. The acting of the template of the new mRNA strand is formed. After elongation, ribonucleotide are added the template strand, they enable to growth of mRNA. After the elongation is over, the termination part also participate. During termination, RNA polymerase encounter the terminator sequence and transcription also stopped. RNA polymerase then release the DNA template. After RNA polymerization, the processing also occurs, that is called RNA processing. The transcript RNA is known as pre-mRNA. The messenger RNA also coder of the protein and also encoding the particular protein in the body. It is processed further converting the mature RNA, and these also include some steps, that is capping, polyadenylation, splicing. And the capping means a methylated guanine cap is added to the protect the mRNA. You know the guanine, adenine, guanine, thymine, cytosine. Everything also nucleotide. Nucleotides occurs in the DNA. During RNA, adenine, guanine, cytosine, instead of thymines, RNA has the uracil. It involves the capping. Addition of methylated guanines occurs, and it occurs the 5' into mRNA transcript. Then it protects the mRNA from the degradation. After that, polyadenylation occurs. So what is the polyadenylation? In polyadenylation means the tail also protects the end of the RNA coded. The mRNA is a degraded. It involves the first one is a endonuclease. It is a cleave the mRNA and specific sequences. Specific sequences means the nucleotide. The enzyme polyadenylation, the polymerase, it is facilitating the addition of several adenine nucleotides. During splicing, the non-coding sequences mean intron removed by the splice some excision. The coding of sequences or any one of the exon is joined together by the ligation. Ligate means it ligates some of the enzyme, it is a pasting. Thus, several number of protein can be made from some single pre-mRNA. After that, the mature mRNA is formed end of the transcriptions. The transcriptions occurs in both prokaryotic and eukaryotic cells also. In prokaryotic cell, it occurs in the cytoplasm. It also have the 3 spaces, the name, initiation, elongation and termination. Normal RNA polymerase also recognizing the strand. The core enzyme also participated and synthesized the RNA, and the normally 5' to 3' to 5' another template also, complementary template also formed. It uses of the antisense strand and DNA as a template the RNA synthesis. Normally, the 3' hydroxyl group is growing and the RNA chain is interact with the alpha, the phosphate group, and incoming the nucleotide to form a phosphodiester bond. The bond also transcript occurs. Some termination sequences, the DNA also formed. When the RNA polymerase to detect the termination sequences, it stops the transcription process. After that, it is releasing and synthesizing the RNA transcript and dissociated from the DNA. After that, formation of the DNA dissociation, RNA new strand also formed. Normally, this transcription is a separate process and occurs in the nucleus. Already we have to know about the prokaryotic cell. Without nucleus, that is called prokaryotic cells. In eukaryotic cells also transcription, it is separating in the occurs in the nucleus. It is 3 types of RNA polymerase enzymes is involved in the eukaryotic transcription process. Normally, mRNA is produced. It has a RNA cap at the end of the pyridation. The polyadenyl is a tail region is formed, and also it occurs in the pyridation. Next, it also have the some RNA type of ribonucleic acid, that is a ribosome to assist the bodies of synthesis of the protein. In the body of the human, nucleic acid is charged to creating the new cells. Everything also is coded by the ribosomes and on some peptide chain. It is often generated from the DNA molecules. The sole of difference between RNA and DNA, that RNA is only one strand, whereas the DNA have the 2 strands. Only one ribose sugar molecules to involving the RNA synthesis. All the cells required the ribosome because it is a main building block of the non-coding RNA. It is called ribosomal ribonucleic acid, that is called rRNA. That is ribosomal RNA. It is also creating some large and small ribosome is subunit. Already it have the small subunit and large subunit. It also have the 3 sites, E site, T site, and also it is ribosomal RNA is translated from the ribosomal DNA. So it is a main role of rRNA and rDNA. The types of rRNA occurs. What are the types? In prokaryotic cells also have the different types. Eukaryotic cells also have the different type of RNA. In prokaryotic cell, 16S ribosomal RNA is housed and it is compared to the 30S ribosomal subunit. The 2 rRNA species are present in the larger 50S ribosomal subunit. This 50S ribosomal subunit we have to divide it 23S and 5S ribosomal RNA. S mean already you know the sedimentation coefficient, that is called S. Therefore, it may be concluded the single rRNA gene, some of the archaea, bacteria, it coded the 3 rRNA, that the type is called 16S, 23S and 5S ribosomal RNA. The bacterial 23S and 16S, 5S rRNA gene are commonly arranged in the co-transcript in the operon synthesis. In this operon synthesis, the eukaryotic ribosome is composed of the 40S and 60S subunit. Now, normally, the eukaryotic eukaryotic ribosome is usually have the several variant of rRNA. It also arranged in the some repeated sequences. About how many nucleotide sequences occurs in 300 to 400 nucleotide sequences. It has the 5 cluster in human chromosome, 13 RNA, that is called R1, R2, R3 and R4. 13, 14, 15, 21 and 22, the number also varied. Furthermore, normally in the chloroplast and the mitochondria in eukaryotic cells, the both containing rRNA, the ribosomal can be found in the cytoplasm. So, it is also free floating complex and connected to the endoplasmic reticulum. Already I told you, all the cell organs are involved in the protein synthesis. This protein synthesis, the primary function rRNA, the adenine, the A site, P site and G site also, it is creating within the larger subunit and connecting with the smaller subunit also. The ribosome is an unusual 3-dimensional structures of rRNA, the internal helix to formation of some loop-like structure. It also used to attaching the mRNA, that is called messenger RNA. After that, it coded into formation of transfer RNA. During that time, the protein also, amino acid also sequentially, they have to connect it. That is called protein synthesis. The sequencing of mRNA approximately translated to the amino acid sequences of all the protein molecules. The tRNA normal transfer RNA, otherwise called transfer RNA, which help in the synthesis from the protein to mRNA. The main function is the adapter molecule during the translation process. So what are the structure of tRNA? The main structure of tRNA, it also has how many nucleotides? More than 70 to 90 nucleotides also occurs. It is a very long, and also each and every nucleotide is specified the amino acid in the specific tRNA. The stop codons, initiation codons and termination codons, everything also occurs. The stop codon means on the time also the sequencing of the amino acid is stopped. After that, formation of new polynucleotide. The stop codon are not recognizing by any tRNA. The secondary structure of tRNA looks to the clover leaf, and tertiary structures is like the inverted L shape of the tRNA. The folded structure is formed due to the hydrogen bonding between the complementary bases also. This is a very nice structure of the tRNA. It also has the arm. How many arms have the? One is the acceptor arm, another one is a D arm, another one is a T arm, another one is a variable arm, another one is a anticodon arm. These also, everything also doing the different functions. Some descriptive bases also occur. That is how many bases? Adenine, guanine, thymine, cytosine. Each and every part also have the 2N. One is a 3-dacian, another one is a 5-dacian. The green color and yellow color and violet color and pink color also they have to indicating the nucleotides. The new anticodons also formed in the anticodon arm. And this sequencing of the protein molecules and amino acid molecule, formation of new number of polypeptide chain, they occurs in the cytoplasm. The clover leaf structures of tRNA function, the folder structure, that is a hairpin loop is otherwise called. It gives the appearance of the 3 normally leafed clover-like structure, and it contains the acceptor arm and the pH loop and variable arm, everything also. The acceptor arm function is formed by the base pairing and 7 to 9 nucleotide and 5 terminal to 3 terminals end also. The terminal have the polyphosphate group and end of the CCA or CCA tail region. After that, the amino acid attaches to the 3-hydroxyl group, and that is arm is called acceptor arm. The amino acylation tRNA charging the tRNA first step of translation process. The enzyme amino acyl tRNA is synthesized and catalyzes the reaction. Another one is a THU loop. The D arm has a stem of 3 to 4 base pair, and the end of the loop-like structure it is called a T loop-like structure. It generally contains a dihydrouridine and modified in the nucleotide. Anticodon loop, it has a 5 bases of it's a long stem-like structure. It also coded a complementary codon, and it has some unpaired bases anticodon. That loop-like pair, the mRNA is coded each codon, and also it specifies the tRNA in the during protein synthesis. The loop-like structure, the T arm also consists of 4 to 5 base pair. The looping contains the pseudo-uridine and modified of the uridines. What is the role of variable loop-like structure or variable arm? It is present between the T-size C loop-like structure. The anticodon loop also contains some various base pair, that is a bases, that is a 3 to 21 nucleotide bases occurs. It helps to recognizing the tRNA molecule to formation of protein synthesis. What is the process of RNA means? Capping and polyadenylation is the 2 types of RNA. It is a process occurs and preparing of the messenger RNA. The protein synthesis, already I told you, some capping region, polyadenylation, and also it coded of the proteins, and also polymerizations occurs. Some enzymes, ligase also occurs. Everything also participate to formation of new synthesis of the protein. Normally, capping region, some special blocking of nucleotide occur. So that is called cap-like structure, and it also have the 59N and mRNA, pre-mRNA formed. During process of the protein synthesis, before that formation of mRNA, that is called pre-mRNA. In polyadenylation, the string of AMP, that is it added the some nucleotide base pair. It may be 3 to 5-N. 39 end of the pre-mRNA it is formed. After the normally transcription, translation, initiation, elongation, everything also formation of new protein synthesis. That is over. After that, another topic is a DNA damage. That is one of the part of the syllabus. We have to know about the DNA. DNA is a double helix structure. DNA also has the coded of the nucleotide. It also hereditary material. It transmits the character from one generation to another generation. Everything also based on the DNA. Without DNA, we cannot live. That is, a DNA is very, very important. So at the same time, some damages occurs in the DNA. Why the damages occurs? What is meant by damages? Damages mean nucleotide changes occurs in the body. Nucleotide changes occurs in the DNA. That is adenine, guanine, thymine, cytosine. But what is the reason to causes of the DNA damage? That is main causes is a mutation. After that the mutation, some abnormalities of the body occurs. Abnormalities means normal hand formation, normal gene formation, normal male or female, everything also totally damaged. Normal DNA damage is the mutation, both type of error in the DNA. Instead of adenine always binding with thymine. Guanine is always binding with cytosine in triple bond. Instead of this one, adenine is not binding with thymine. That time also genes also vary. The DNA damage is the physical abnormalities of the DNA. The single or double strands also break down. After that, the mutation occurs. Mutation occurs, the changes of the base sequences of the all the DNA is varied. The mutations occurs in the mutation with the sequence of the bases of the DNA. They can be large scale, altering the structure of chromosome. The small scale, where they only after the few or the single base nucleotide occurs. Now, not why the reason of the mutation occur. For example, DNA mutation can be caused by the some mistake, some of the polymerase enzyme during replications, everything also process is a formation. The DNA mutation can also result the replication of DNA. They has been damaged. So it may be occurs in the endogenous or exogenous. The DNA polymerase encountering the damaged DNA and also template to replicate the random nucleotide across the lesions also occurs. Some mutations also occurs. After the formation of DNA damage, the environmental factors also involved some metabolic activities. The rate of the metabolic activities occurs in the inside of the cell. That time also all the molar and the molecular everything also changes. Yeah, the changes mean base pair also changes. Approximately human genome have the 6 billion base pair. If left unpaired, it is a repaired, the mutation occurs or any one of the changes, base pair changes approximately, it increases some of the syndromic disease. Sometimes it may be the formation of bulging of the tissues. Due to this, some tumor also formation. After the tumor is leads to the cancer. The vast majority of DNA affect the primary structure of double helix structure and comes up to the some chemical modification occurs in the body. It interrupts the molecule and addicts the double helix structures. Unlike some protein and RNA changes also occurs, it usually tertiary structure. Therefore, some damage and disturbance occurs in the some histone and some structures and effect of DNA, everything also. What are the process in some environmental exposure of the DNA damaging? Some of the agents also causing the damage. First one is a oxidative process. Another one is a alkylation process. Another one is a base loss of caused by the some hydrolysis of the bases. Some bulky adduct formation. Some of the DNA is cross linking and DNA strand breakout and including some single stranded DNA and double stranded DNA to break up. These are the damaging the causing of the some agent. The DNA damage also leads to the formation of new organs and formation leads to the some cell death also. Another topic also apoptosis, another one is synthesis. Apoptosis and synthesis are both the cell process that can help to prevent the cancer. They have different characteristics and effect on the body. Apoptosis, the generally programmed process, it remove or damage to the unwanted cells. In our body, some apoptosis is triggered, some extinct and death receptor, some of the instinct of the mitochondrial pathway. The apoptosis, very good example is a frog stages. During frog, the larval stages also have the gill and tail region. After that, they have to developing. The gills also changed, the respiration also changed, the tails also modified. That is cutting to the frog. After that is cell cessation. A metabolically active process that causes a cell to age and stop dividing but not die. The cessation cell to secrete the number of factors. It may be promote a growth that is called tumor growth or invasion. After that, the tumor growth also formation of DNA repair. The DNA repair means we have the repairing of the DNA after the formation of new things. It also occurs in the some formation of finding of involving of some enzyme. The cells within organism needed to have the ability to maintain and conserve the DNA sequences. The cells also doing the normal function of the organism. After that DNA repair, how the cell give the correct response and doing some function and also alternations. Normal organisms, it depends upon the genetic information. It also coded the DNA and survives and reproduce to make the crucial and also maintain the integrity of the molecule. This is important to the even the DNA damage cannot be repaired, but the following some issues might be happen. It is because some of the cell totally changes, the body also effectively changes. Normal the DNA repair affects the function of survival of the somatic cell. Somatic cell means normal body cell. During fertility cell, the reproductive cells can get adversely affected. What are the causes occurs? Some gene mutation occurs. During the gene mutation developed the tumor cells. The DNA damage repair is most causes and also restore the structure of DNA, but at times it might not be eliminate the DNA damage, yet it allowing the tolerating the DNA damage and through the repair and genetic stability, and also it is guaranteed by the very crucial and reproducing the defect of repair. Some mismatch repair excisions, that some of the part also involving the mismatch and repair. That is mismatch mean in case any one of the cells, the genes, the nucleotide is mismatched. That is called MMR, capital M, capital M, capital R. The DNA mismatch repair is involving the repairing of the correct mismatch of DNA molecule due to the some insertion and deletion of the nucleotide. When it is occurs during the replication process. Insertion means the inserting of the another one of the nucleotide in the particular places. Deletion means deleted of the adenine, guanine, thymine, cytosine, the deletion of the any one of the nucleotide. Another excision repair means this is present in all type of biological cells or living cells. The primary DNA repair mechanism in human cells, it is mainly target is a excision repair or nucleotide in the bases. If we say excision repair, the base pair of the removal of some damaging, that is a glycolysase and hydrolyzing of the glycosidic bond. Normal nucleotide excision repair is targeting some dissociation of the DNA double helix structure, such as it is mainly the any one of the thymine, some dissociation and any one of the addiction, everything also occurs in the base pair. Another one is SOS repair. This SOS repair, the DNA repair occurs in the cell or in the critical state due to the several DNA damages. This type of repair can only to improve the survival rate of the cell and maintain the some gene integrity. The DNA repair also called error-prone occurs in the cell since many mistakes are still left after SOS repair. This repair, some easy to cell to mutate to after SOS repair, formation of new cell in the particular places, they give the some of the reasoning of the SOS repair. In this RNA synthesis process, we have to raising many questions. What is the process of transcription? Another one is here, where the transcription is started and where the transcription is terminated. What is the end product of the transcription? What is the main question? Intron and extron. Normal non-coding sequence of the gene is called intron, whereas the coding sequences is called the exons. The intron and exons also very important of the RNA synthesis and formation of these things. Thank you.